Hello, everyone. I'm Susmita Sahoo from Icahn School of Medicine, Mount Sinai. And I have the pleasure to chat with Dr. Wu from Stanford University, who do not need any introduction. And we just had the session on emerging cardiovascular translational technology in the morning. And thank you so much for your educational talk today on the very contemporary and topic on stem cells, genomics, AI, and drug discovery. And I'm curious, what was your inspiration behind the research and which is revolutionizing our approach to drug discovery nowadays?
Well, thank you, Susmita. It's good to talk with you. So as you know, I'm a physician scientist, and I've always wanted to see patients and understand what the problems are for some of the diseases they have. Go back to the lab and try to figure out what's going on. And to be able to do all these at different steps, you got to bring in different players into the room and have them all play together. And in this case here, the players are the different technologies. And so, as I mentioned in the talk, we do a lot of genomics sequencing. We do a lot of stem cells organoids to be able to recapitulate and capture the disease that the patient has. We also do a lot of drug screening and then drug development. And once we develop the drug, then we go back to the stem cells from the patients to test whether the drugs work or not. And if the drug doesn't work, then we tell the AI software that, hey, the drug doesn't work. Can you further refine your prediction? And then we do it again and then again to further improve the process. Yeah. So at the end of the day, we want to be as translational as possible so that the research that we do in our lab translates to the patients who are sick or at home. Yeah.
Yeah, thank you. And as we all know that nowadays both the NIH and the FDA provided important guidelines towards adopting this cutting edge technology for a non animal research approach. Could you please describe what are the steps? Because for lay scientists and person like us who are not involved with clinical trial, it is important to know which steps comes first, the animal testing because it's a physiological setting, or the clinical trial in a dish. What is the
Yeah.
steps,
Yeah.
how we approach that?
So I think back in April this year, the FDA came up with the announcement about NAMS. It's NAMS. It's essentially stands for New Alternative Methodologies, and this was followed by the NIH advocating for more human relevant based research. In a nutshell, I think in the past we've been doing a lot of research on mice models, transgenic mice, for example, knocking out different genes. But at the end of the day, mouse and humans are not the same, right? Mouse heartbeat is like 600 beats per minute.
Yeah.
Human is 60 beats a minute. A lot of the genes that are present in a mouse does not exist in human and vice versa. And so you may develop a drug that works perfectly in a mouse, but it doesn't work in humans. And so one way to think about this is to turn it around and say that now that we can generate stem cells from pretty much all our patients, would it be possible to grow these stem cells from the patients, test whether the drug work or not? And then once you're confident that the drug work, you can then still take the drug, go back to the mouse, and just make sure that there's good absorption, there's good distribution, excretion, and metabolism of the drug.
Yeah.
And that's essentially the pipeline that we set up in our lab. We do most of our drug screening first in human relevant based system. And once we like the drug, we test it in our mouse models. Yeah.
Yeah, that makes
And
sense.
yeah, at the end, I think the other advantage is it reduces our reliance on animals. Yeah.
Yeah. Thank you. And I'm kind of curious, has there been any drugs that is in the FDA pipeline or that has been already approved that, what are the success stories from that? And how do we look forward to developing and using these kind of drugs in patients?
To my knowledge, there haven't been any quote unquote drug that have been approved by the FDA. To my knowledge, there are a lot of companies and a lot of labs that are doing this already. I show you the example of what we've been able to do by using genomics, stem cells, and AI to repurpose genestin. That's in clinical trial phase 1 to repurpose artesunate. That's also got the IND approval for phase 1 trial for patients with idiopathic pulmonary fibrosis and a separate trial for pulmonary arterial hypertension. Yeah.
We look forward to knowing the results from this. And what is your key take-home message? And what do you think could be the long-term impact? And how do we handle failure if there is any in this approach?
Yeah, I think it's a very important question because at the end of the day, it's very cost prohibitive to develop a drug. I think the common numbers that people will quote you is it takes $2 billion and it takes about 10 to 15 years to take a drug from the R&D stage, research and development, all the way to the clinical trial. And if you're a drug company, drug 1 fails, drug 2 fails, drug 3 fails, drug 4 fails. By the time drug 5 succeed, you want to recuperate all the costs from drug 1, 2, 3, and 4. The way you recuperate the costs is get the price high, and then the consumers complain that how come this drug is so expensive. I think if we can have a better success rate for developing drugs, hopefully the cost will be lower. It's more affordable to all of us. And then using these human relevant models, hopefully the drug will also be more efficacious and safer. And that's the goal, and that's what we've been trying to do in our lab.
An exciting future. And thank you, Dr. Wu, for
Thank you.
joining us.
Yeah.
Thank you, everyone.
Thank you.